Hello, my name is Lauren Edwards, and I would like to first thank you very much for considering my proposal. I'm going to tell you a little bit about myself first. I'm a graduate student at the University of Massachusetts, and I'm studying industrial mathematics. I've always had a fascination for biology and mathematical applications to biology. In my senior year at WPI, I got to learn how to make a good mathematical model. This project that I did actually has quite a lot to do with the proposal I am suggesting. So I'm going to talk a little bit about that first, and then I'm going to go ahead and go more in depth about the proposal. During my senior year, I completed a project that focused on Epstein-Barr virus models. Specifically, I looked at one that displayed the interactions between the virus, its host cells, and our immune system. This is a general overview of that model. While Epstein-Barr virus is known to cause mono, it's also linked to many other illnesses. This stems from the fact that it can infect two different types of cells, B cells and epithelial cells. One main theory and assumption of this model is that Epstein-Barr has two different shapes. They are still the same virus, but each shape can more easily infect one type of cell. As you can see, each virus shape fits one type of host best. The better the fit, the better the chance of attaching and passing genetic material into the host. When the virus enters the lytic cycle, those forming in the B cells have the shape that will best fit the epithelial cells, while those forming in epithelial cells will fit B cells best. These cells burst through the cell wall and proceed to look for a new host. While most viruses attach to a host cell with the same shape, you may notice that this is not the case for all of them. There is always a small chance that a virus that is not the same shape will attach. These graphs show the numerical results of Hund's original model alongside the simplified model that I produced. As you can see, each model produces similar numerical results as the original. And the most important part, the steady states are the same. As you can see, the solid line represents the numerical results of the original model, while the dotted line represents the results of the simplified model. The research that I am proposing will explore a potential treatment option for Nasopharyngeal Carcinoma, or NPC, which is one illness the Epstein-Barr virus has been linked to. NPC is caused by the proliferation of epithelial cells, and has been found that these cancerous cells contain copies of latent Epstein-Barr virus. This treatment option involves inducing the lytic cycle in these latently infected cells. This would cause the virus to replicate and then lyse the cells. Since the late infection is only found in cancerous cells, it would be a very direct treatment that would leave surrounding healthy cells relatively unharmed. But this raises a few questions. What percentage of cancerous cells must be induced for this treatment to be effective? What are the potential side effects? Assuming the virus that's released is the shape to infect B cells, would sudden increase of free virus cause some other illness from increased infection of B cells? Mathematical models can serve two main purposes. The first is to verify a theory. Does a model based on theory yield similar results to in vitro results? If so, the theory is supported. This is what Hund did with the above model that I described. Models can also be used to predict, as is done with the flu vaccine. A model is made to predict the future genetic changes of the flu, and thus a vaccine is created. Through building a mathematical model, I hope to analyze this model and obtain potential answers to a few of the above questions, if not all of them. Knowing these answers would greatly help any future investigation on this treatment. Again, I'd like to thank you for your consideration and your attention.